Good afternoon, everyone, and thank you for joining us today. My name is Erin Stagletter. I'm the Education Manager for Virginia Breast Cancer Foundation, and we are embarking on a new adventure for VBCF, educational webinars and Facebook live streaming. This webinar will also be recorded, and we will plan on making it available on our website at a later date. Today we have Dr. Luisel Ricks-Santi from Hampton University to speak with us today. She's the director of the Cancer Research Center at Hampton University and an associate professor in the Department of Biological Sciences. She received her PhD from Georgetown in tumor biology and postdoctoral fellowship at Howard University in cancer genomics and cancer disparities research. We're so lucky to have her with us today. And my colleague Nikki Jennings will be watching Facebook for comments and questions as they come up, and I will be watching Zoom for those questions. So we'll be able to be interactive with our audience. So with that, Dr. Ricks-Santi, you are welcome to begin.
Thank you so much for your invitation to the first, what I hope is of many presentations to ensure that our community is well-versed and educated on the cancers that are unfortunately affecting Virginians. And so I'm going to go ahead and share my screen. And hopefully you can see my screen. Erin, can you see my screen?
Yes.
And so my discussion today is called Educating Communities About Breast Cancer. And I was just mentioning to Erin that because I'm the first, I can kind of give an overview about breast cancer and how it's affecting Virginians. And so I'm happy to be the first. And then again, there's nobody to compare me to because I'm the first. So I really thank you for this invitation. And so today I'll talk about, I'll give the community broad strokes about what breast cancer is and how it's described and how it's characterized, just to give us some, an index and to orient our audience about breast cancer. And so first I'd like to qualify why I'm doing this talk. So I actually graduated from Hampton University, received my bachelor's from Hampton University in molecular biology. I went to Georgetown, as Erin mentioned. I got my PhD in tumor biology, received a couple of grants, went to Howard, did my postdoctoral fellowship in cancer genomics and cancer disparities. But then about 7 years ago, after the opening of the Hampton University Proton Therapy Institute, was invited to return back to my home by the sea and begin and promote cancer research here at Hampton University. Our Proton Therapy Center, as most of you may know, is a cancer treatment center. However, my area is research, and so the goal was to expand our research portfolio, apply for grants, and engage more minorities in health disparities research, including clinical trials. And so when I tell people that I received my degree in tumor biology, you can imagine that the first question they ask me is, well, when are we going to find a cure for breast cancer? When are we going to find a cure for cancer, period? And so, you know, I often have to explain to people that cancer is not just one disease. It is the type of cancer you have depends on the organ that is affected. And then within your organ, you have different tissues. And then within the tissues, you have different cells that have different functions that express different proteins. So cancer is actually not one disease. And even within one organ, you can have different types of diseases, including for breast cancer. So I'm going to talk a little bit about how breast cancer can be described and how it can be classified differently. And because of its differences in classification, will require different forms of treatment. So one of the first ways that breast cancer specifically, and cancer in general, is classified is by staging. And staging has to do with how far the tumor has spread and how large it is. So many of us have heard of stage 0 breast cancer. And so that is carcinoma in situ. And what that means is that the tumor, or these little bunches of tumor cells, have not expanded beyond certain margins. And so that's stage 0. And the survival rate for stage 0 breast cancer is around 100%. Then there is stage 1 breast cancer, which has a 98% survival rate. And again, this is a tumor that's a little bit larger. It's a little bit more invasive, but it has, again, not migrated beyond its borders. Stage 2, they get a little bit bigger, and there is a little bit of migration and invasion, potentially into the lymph nodes. Then you have grade 3, which again, there is a little bit more spread and migration. And stage 4, the tumor has spread from the breast to different parts of the body, to include the brain, bones, lungs, liver. So it can spread to other areas of the body. And so what I like to tell people is that breast cancer, there is a cure for breast cancer, and one of the best cures is prevention, but the next best cure is to find it early. Because when you identify cancer early, in its earliest stages, it's still confined and it has not spread. And the reason it's easier to treat also is because once it spreads, in order to spread, the tumor has to change. So the tumor that migrated from the breast to the brain is no longer the same type of tumor that is in the breast. So you're no longer treating just breast cancer, you're now treating breast cancer plus a metastatic lesion that's in the brain that is different than the original breast cancer. And then another way to classify breast cancer is by the way that it's graded. And I like to say that this has to do with how ugly or how pretty it is. So the breast itself, the function of the breast is to produce milk and to excrete milk. And so you see these white holes, those holes or those circles are where the milk, and those are the ducts, and that's where the milk actually gets excreted into. And so in a grade 1 tumor, it still looks like the breast. It's still very pretty. You can still see the structures of the breast. In grade 2, you start seeing those circles, those white circles, those ducts get filled up with cells, but you can still kind of tell that that's where you're supposed to have milk. But in the grade 3, you can no longer distinguish the ducts from the rest of the breast tissue. And so grade is also associated with survivorship, with grade 1 having a better survivorship than grade 3. And please feel free to ask any questions in the chat or out loud. I'm happy to answer them. And as I mentioned, even the breast is made up of different tissues with different functions. And so I'm not sure, can you see my arrow or my laser? Well, there's a laser pointer. Can you see my laser? Excellent. So this here is a breast, and then you have these ducts here. And then I like to call these little lobules little grapes. And so the grapes have a function, the ducts have a function, and even in the cells that line the lobules, you have different types of cells. You have basal cells, you have epithelial cells, and then here's the lumen where these cells can excrete milk, and they all have different function. So not only can you get breast cancer in these epithelial cells, you can get breast cancer in the basal cells, you can get breast cancer in the lobules, and you can also get breast cancer in the ducts. And then those are all very different types of cancers. And in fact, most breast cancers tend to be ductal carcinomas, and very few are lobular, but there is an association of lobular cancer with worse prognosis. And so even within the tissue, even the type of tissue breast cancer you have can determine how you should be managed and treated. Breast cancer and breast cancer cells can also be described by their molecular subtype or the proteins that they're expressed. We'll focus here on 3 particular proteins, and just imagine that this is a cell, this is your nucleus, and most breast cells express these little antennas, which are receptors for hormones. So this is a little antenna on the outside. You also have these little antennas on the DNA that will also bind hormones like estrogen. And depending on whether you actually have those antennas. So in some breast cancers, you actually lose these little antennas and ways to receive signals. And so you can imagine that losing the HER2 receptor and the progesterone receptor and the estrogen receptor, that is why we have a type of breast cancer called triple negative breast cancer, because what that means is that you've lost these little proteins, these little receptors that receive signals and tell the cells to start growing or to stop growing. So this is another way that breast cancer can be classified. And so here I'm showing that when you lose these receptors, your cancer can look very different. And because those receptors are also targets for treatment, the treatment of a tumor that has those receptors versus the type of tumor that doesn't have the receptor, the treatment for that is going to be very different. And so molecularly, we have several subtypes. We have some breast cancers that are estrogen receptor positive and progesterone receptor positive, and those are called luminal breast cancers. You have those that are ER negative or estrogen receptor negative and progesterone receptor negative, and they can be either basal-like, but if they express the HER2 receptor, they can be HER2 receptor positive, and here if they're triple positive, they're called luminal B. And this is important because, again, these subtypes are also associated with prognosis, and I think that I have a picture of why they're important for treatment. So tamoxifen is a drug that is often prescribed to patients that have the estrogen receptor. And what they do, so these little pink dots here, imagine this is the cell, this is the nucleus, this is the DNA, and the green hook right here is an estrogen receptor. Tamoxifen blocks those receptors and prevents the division of cells. So instead of promoting division, it blocks division here. And Herceptin is very similar, but Herceptin is on the outside of the cell. And there is, HER2, the HER2 receptor is on the outside, and there's a drug called Herceptin that will block those receptors on the outside of the cell, which again will inhibit the cell from growing, and it will stop it from growing and dividing. So again, you can imagine if a patient loses those receptors, then there's a limited number of ways that one can treat that patient.
Dr. Ricks-Santi, are you going to be getting into breast density at all?
I will not talk about breast density today.
Okay. Well, I did have a question from one of our audience members, Sabrina. What about dense breast? Does that have an impact on the type or grade of cancer someone can have?
You know, there's been a lot of research going on and trying to decipher what the impact of dense breast, and I think that research is coming out now. I think that it's important to understand the composition of the density, and now we're starting to understand the role of fat, and there are different types of fat and the amount of breast tissue, because we're starting to understand now that depending on the kind of fat and how much fat you have, there are certain hormones that are being released that could impact, again, how breast cells respond to their environment. So that's still an area of research that is gaining a lot of traction, but it's moving very fast, and I think we're starting to understand how breast composition may impact. And I think that there is a correlation between having dense breasts and breast cancer risk, and I think some of that connection may have to do with our reduced ability to diagnose cancer earlier, right? So women with dense breasts, instead of getting a regular mammography, there might be ultrasound that may be recommended. So again, the correlation can be with the fat cells promoting cell division, or it can be the fact that we have a limited imaging capabilities to be able to diagnose breast cancer early in dense breasts.
Thank you.
So my answer to the question, have we found a cure for cancer, is absolutely. Have we found a cure for all cancers? Well, it depends. And so most stage 1 breast cancers are treated with surgery or radiation therapy and sometimes hormonal therapy. And now you have immunotherapy that's emerging and is very promising. But it's all going to depend on your staging, on your grading, what molecules are being expressed in your tumor. But there is some correlation with the type of surgery that you get and the state and your staging. But all of that will be very personalized by your physician, depending on the molecular subtypes and some of the other characteristics that I've mentioned. So I won't go into that. So again, I want to reiterate that early detection is the key. And in fact, when you catch breast cancer early in stage 1, survivorship is more than 98%. And so that's why prevention is key, and that's why mammography is key, and that's why as women we fight to be able to get our mammograms at 40 years old. But also we need to understand what are some other risk factors that can put us at risk for some of these other high stage breast cancers. And so one of the things that I study here in the lab is cancer disparities. I am particularly interested in why African American women, while the risk or while they're at the same, they have the same rate as white women to get breast cancer, their mortality rates tend to be much higher. So here you see white women and black women, and their rates are very similar, much higher than for Latinas and for American Indian and Asian, but they're very similar. But then you see here a spike in the number of deaths that you see attributed to breast cancer in African American women. So I'm really interested in understanding, why do we see this spike? Is it molecules? Is it genetics? Is it the environment? I'm particularly interested in molecularly, what is it, what type of breast cancers Black women tend to have, and I tend to look at those characteristics to help me understand how much those characteristics contribute to this disparity. And so in some, I think this is data from the state of Virginia. Actually, I don't know, this might be from Washington, DC. When I was in Washington, DC, I was looking at the differences in stage in Caucasians versus African Americans, and ER, estrogen receptor expression. Here I was looking at grade, and here I was looking at the types of breast cancer. And so we see here that African American women have a much higher rate of stage 3 and stage 4 breast cancer. African American women have a lower rate of estrogen receptor positive breast cancer. They have a much higher rate of high-grade breast cancer, and they have a much higher rate of triple-negative breast cancer. So these are the things that I'm actually trying to understand using molecular and genetic methods, so we can come up with better ways to improve the disparity or to reduce the disparities among these groups. And so as I mentioned in my figure, triple negative breast cancer is a type of breast cancer that is missing the little antennas that I showed you earlier. It's missing all 3 antennas, which are targets for treatment. And in fact, triple negative breast cancers account for approximately 15% of all breast cancers. So across the spectrum, doesn't matter what race, they account for 15% of breast cancers. These are very difficult to treat breast cancers. They tend to be associated with higher grade breast cancer, again, because they don't have those hormone receptors, the estrogen, progesterone, or human epidermal growth factor receptor. They tend to be unresponsive to hormonal therapy like tamoxifen and Herceptin. There is an association with survival, and they tend to have a poor prognosis. And depending on where you are, in Washington, DC, I found out that African American women had about 30% of African American women had triple negative. But I have seen statistics elsewhere that have shown that African American women comprise, or that African American, within the group of African American women, triple negatives comprise 50% of the types of breast cancer in that group. So triple negative breast cancer could be accounting for the differences in mortality between Caucasians and African Americans with breast cancer. As I mentioned, I'm very interested in the genetics of breast cancer. And so one of the things that we have noticed in time is that triple negative breast cancer is also a disease, or it's a type of breast cancer that is found in families and in BRCA mutation carriers. And so I show you here what we call a pedigree. The circles are women and the squares are men. The lines indicate that that particular person has passed. Red, if you see a red quadrant, that person had breast cancer. Green quadrant, that person had colon cancer. Yellow quadrant, that person had lung cancer, and blue quadrant is another type of cancer. And so this is all to show you. So this was a young lady that came in at 73 with triple negative breast cancer, and most of her sisters and her brother had been affected with cancer, and several of her sisters had passed. But her mother had passed of breast cancer and her father had passed of colon cancer, yet this young lady right here came up BRCA negative. She was an African American woman. She was one of our patients at Howard University. And so this was the inspiration for the research that I'm currently doing. And so as I mentioned, dysfunctional or having mutations in BRCA1 and BRCA2 increase the risk for triple negative breast cancer. And it also increases the risk for ovarian, pancreatic, and prostate cancer. But what we know about BRCA1 and BRCA2 tends to be limited to Ashkenazi Jewish women or white women. And so specifically BRCA1 and BRCA2 are responsible for helping DNA repair itself when it has mistakes in it. And there is genetic testing and genetic counseling available. And so my question, given my interest in health disparities, was Black women have a high incidence of triple negative, and triple negative is associated with having what we call BRCAness, and that's deficiencies in DNA repair. So what do we know about BRCAness in African American women, or what is the impact of BRCA, if any, in African American women? And so while I was at Howard, we were able to collect blood samples from patients with breast cancer and their family members that were affected and not affected. We also collected clinical data. And me, I'm actually a bench scientist. So what I really enjoy is being in the lab and doing what you see right here and analyzing data. So I was able to analyze the data from these families. And unfortunately, for this group of African American women, we found that we looked at BRCA1 and BRCA2, but for 60% of the results, or for 60% of the women, the results were inconclusive. So even if we saw something, we would not be able to counsel the patient and say, you know what, we saw a positive result, or you know what, we saw a negative result, you're good to go. And same thing for BRCA. And so often, and what you may or may not know is when you have a family history of any type of cancer, you may get recommended for genetic testing or genetic counseling. And so you'll go in, you will either donate a saliva sample or a blood sample, and they will do sequencing. They will either look at 1 or 2 genes, they could look at a panel of genes, 60 genes, or they can look at all of your DNA. Your results come back, and for some women, it says, you know what, this is the gene that is causing breast cancer in the family. It's conclusive. We know, and because we know, and because you have family members that also came back positive, we recommend some prophylactic surgeries or preventive medicine, so you can manage the disease to try to reduce the risk in that population or in those family members. However, if your test comes back inconclusive, it's very hard to make recommendations to that family because we don't know what the results mean. And what we're seeing more often than not is that Black women or women from different ethnicities other than Caucasian women have more inconclusive results, which makes it really hard to manage breast cancer or different types of cancer in that family. And so this was a study that was funded by the National Institutes of Health. And the project was to study the genetics, how genetics impacts breast cancer in African American women. But another really important part of my research was to understand, and not only understand, but improve the conclusiveness or the conclusivity of inconclusive results. So if somebody comes back with an inconclusive result, okay, give me that inconclusive result and let me see if I can make an experiment in the lab and determine in the lab in a petri dish whether changes in that gene at that particular area results in changes in the behavior of the cell. So my job through this grant is to functionalize these inconclusive results. And so lastly, I want to get into, well, second to lastly, I want to get into clinical trials. These studies would not be possible without clinical trials. And even me as a scientist, as a seasoned scientist, when I think of clinical trials, I often think about being injected with something and then watching me through time. And what I like to tell people now that I know better, is that clinical trials can include observational trials or interventional trials. Interventional trials, yes, you may be injected with something like the COVID-19 vaccine trials. That's an interventional trials. But there are things like observational clinical trials, which do not test drugs. They will observe you over time. They may ask you to donate a saliva sample or a blood sample and maybe some of your information with the goal of trying to understand the disease. But the goal of the observational trial is not to inject you with something and then watch you. It's really for you to make a donation of data or blood or something like that, and then for us to make connections about your genetics and the disease in your family. And so what I do a lot of is genetic trials, and the goal of the genetic trials is to improve the prediction of what your genes are doing. And so again, we could not determine conclusively what your result means unless we have a group of women with similar results, or unless we can recapitulate or repeat what's going on in your body in a petri dish or in an animal. And so genetic trials are important, and engaging in genetic trials are important for us to improve the conclusivity of tests, of genetic tests. And the more data we get, the better we get at telling you what the data means. And so at the very beginning of BRCA testing, there was a lot we didn't know, but we know a lot more because more and more people are donating their information, are engaging in genetic trials, and are engaging in clinical trials. And so genetic trials also can lead to the development of tailor-made treatments. Where we're going now is this area called precision medicine. And so the right medicine at the right time for the right person. So if you have a certain genetic makeup, we may be able to develop something in the lab that's specifically for you. However, we need people to engage in these genetic trials. So there's currently a big trial at the National Cancer Institute. It's called the MATCH trial. And what they do is they ask patients to donate their tumor. Usually a pathologist will take a look at their tumor, and after they're made their determination, it either gets stored away and some of it even gets disposed of. But what NCI is doing is asking patients to donate the remnant or the remaining tissue into a biorepository, and then they're trying different drugs on that tumor to determine the best course of treatment. And so again, this is not an interventional trial because they're not injecting you with anything, but here is your opportunity to make a difference in the amount and the type of knowledge we know because of your donation. And so here's a genetics guided trial. So this was a gentleman that had melanoma, and this is a bone X-ray, and everything that's black is essentially, besides the bladder, is a tumor. And so they tried a specific drug that targeted a specific gene mutation And after 15 days, you saw almost loss of, or not loss, but cure of the tumors in his body. And so through this trial, I found out that, now this here is an interventional trial. This is an interventional trial. So he was a stage 4 patient because it had migrated from, I don't know what kind of melanoma, but it was all over his body. And then here, they were able to look at the tumor and look at the mutations in the tumor, and there is an actual drug that targets that mutation, and we're able to treat him and see how he responded to the tumor. You may or may not have heard of the All of Us program, the Precision Medicine Initiative program, and this hopefully is a program coming really soon to the state of Virginia. And again, the goal is to look at everyone's environment, lifestyle, and their genetics and biology to help understand how your genetics and how all these things play a role in whether or not you get sick, how healthy you are, how you respond to different drugs. And so this is, I'm really interested in being engaged in this All of Us trial and helping with the engagement of patients and of community members, because you know it's going to take all of us to be able to give enough data that will benefit the population as a whole. And why is it important for more people to volunteer for trials? And so these studies are very, very important in the development of new therapies. So you can imagine, you know, even at the beginning of a trial, we understand that 85% of clinical trials fail to even get enough patients for the clinical trial. And then 80% of trials fail to finish on time. 50% of trials enroll 1 or no patients. 40% of trials, most of the budget goes towards recruitment, and many patients drop out of clinical trials. So there's a great need, not only for more patients to enroll and to engage in clinical trials, but more diverse patients to be involved in clinical trials. And here, 3% of cancer patients are actually participating in clinical trials, but we find that around 85% are unaware that they can participate. And while I was at Howard University, I had a graduate student do a study on why patients did or did not participate. And what we found was that it's because they didn't know. So it's not because they didn't want to participate, it's because a lot of people don't know. So this right here was very true in our particular population of patients, which was mostly African American. It's not that they don't want to participate in the trials, that nobody really approaches them about the opportunity to participate in a clinical trial.
I have a question. So, obviously this is what you do. So, what strategies have been helpful for you in the past in being able to recruit participants, recruit a diverse group of participants, or do you have a success story that you can tell that you tried something and it worked and it was wonderful?
So I think there's a couple of things that I find work very well. You want to ensure that you have recruiters, nurses that are congruent and look like the people that you're trying to recruit. That's important. Another thing that I find is that you can't be robotic in your approach to people. You can't just have a piece of paper and you're reading, right? This has to be consent. There's a requirement for informed consent. And to me, informed consent is a conversation. It's not, I need to read this for you and you can't understand anything. It's, well, let me tell you about this. And let's have a discussion about why it's important and why it's not important. And you know what? You have time. You can come back later and then we can talk about it. Here's my number. So being personable, understanding that there is a person in front of you and it's not a race, it's not just don't be robotic. I think another good approach is making sure that the doctors and the physicians and the clinicians are also bought in, and that even the clinicians see the benefit of patients participating in a clinical trial. So there's several things, and I think that, do I have a story? I have lots of stories in there. Actually, I have one not successful story, and it was because it was a gentleman that was scared of needles. He said, I'm scared of needles. I don't want to donate blood. And so I totally understand. I said, but when you're ready, I'm here. And, you know, I thought it was interesting. I thought it was funny, but it's a true fear. It's a real fear, you know, and you just have to respect that. So hopefully that answered your question.
Yeah, thanks.
Mm-hmm. So how do you get involved in clinical trials? And so when folks ask me, where can I go to find a clinical trial? I often send them to clinicaltrials.gov. And literally you can go to clinicaltrials.gov and you can look up any health study. You know, you may be interested in contributing to an obesity research study. I am actually part of with the National Eye Institute because I have a genetic disorder that affects my vision. I'm part of a clinical trial at the National Eye Institute. And they essentially, it's an observational trial and a genetics trial, and they follow me over time. And I was able to find that trial through clinicaltrials.gov. So you can go into clinical trials and you can search by type of cancer, you can search by state, by city, by lots of different things. It's a really cool and very easy to navigate search engine. But if you are looking for somewhere or a trial to participate in, this is a great start. In addition, we need healthy volunteers. So a lot of these studies need healthy volunteers. So if you're looking to contribute to science, to make a little extra money, I don't know, I did that in college. I make a little extra money. I was a healthy volunteer. You can go to clinicaltrials.gov and you can find lots of different opportunities to engage in clinical trials. And so lastly, I want to take some time to talk about Family Health History Month, which is November. Family Health History Day is Thanksgiving. And I'm doing this now because tomorrow it'll be Christmas and we'll forget about the way time is just passing us. I just want to create some awareness about family health history. And family health history is a combination of not only the genes that we share, but our environments, our behaviors, and our culture. Family health history is information about our surroundings, our habits, and those of our family. And it's a powerful tool, though. So if you ever are recommended for genetic counseling, you're going to have to collect this information. And so we're going to ask you information about the behaviors in the family, the environment, the cultural factors, how you cook your food, how your family cooks their food. All this information goes into your family health history, which can help establish which is the best test for you or for your family members. And we all have a family history of something. I mean, there's diabetes in my family, high blood pressure. And because this is something that I'm very aware of, often when I get together with my family, I'm asking questions. So when my eye condition came up, I said, all right, guys, so who else has this eye condition? What's going on? I heard this is, genetics, and those people started talking about somebody had cataracts and somebody had this, and did you know that your grandfather had the grayest eyes? And did you know that so-and-so had curly hair? And so you start here, but then you get so much information about the family that could be useful, especially if you have a condition like cancer, which can be helpful in how you are managed and how you are treated. And so the family health history is important. It can tell you about your personal health. It can guide screening. So one of the most important things that I like to tell people is that breast cancer or mammography starts at 40, right? For most of us, it starts at 40. But if you had a case that was much earlier in your immediate family, you were supposed to go at least 10 years before that first case. So if you had a case of breast cancer at age 40, your mammography, or you should start screening at age 30. And so this is information that you should share with your doctor in case just to be able to make decisions or be able to manage when to get those screening tests. And I saw a hand. I can't hear you, Erin.
Sorry, I just realized I was muted. We did have another question. It was more tied to the clinical trial aspect, but if someone is interested in typing a question into the Q&A box or the chat box, either one will work for something currently related to the family health history discussion. We can tackle that.
Okay.
I think we should just wait until the end to revisit the clinical trial question.
Okay. All right. Sounds good. Importantly, you know, when I found out that I had a genetic eye condition, I started to get worried about my daughter's eyes. And so now for me, it's really important that she makes her optometry appointment. I mean, how many of us skip our optometry appointment every year? I skipped it for 10 years, more than that. And so now I make sure that my daughter, at least my daughter, goes to her optometry appointments, because if there's anything that I can do at that time, then we can try to do that. So it's helped me take control of my own health. It's helped me take control of my daughter's health. Also, I have a family history of diabetes. And, you know, about 10, 15 years ago, I told my mom, you know, my HbA1c levels are really, really high. And you know, what's going on? And she's like, oh yeah, so-and-so, your grandmother, your grandfather. I'm like, how come I didn't know any of this? And I didn't imagine it in my family because I didn't see obesity in my family. And so I didn't think it was something that was going to affect my family, because I associated diabetes with obesity and some other lifestyles. But because of that, I was able to go back to my doctor and we were able to get a nutrition plan together to help me take control of my HbA1c and my sugar levels. And so family history can actually be a risk factor. It's a risk factor for cancer, it's a risk factor for diabetes, and it's a risk factor for cardiovascular diseases and many other diseases. So knowing the family history, it's really the best way to help you learn about and reduce your own risk factors for disease. It'll help you become more aware of your health and the health of your loved ones. And importantly, this information can help you lead a healthier lifestyle. And like for me, knowing and understanding that diabetes ran in my family helped me, and it potentially saved my life, because now I know that I'm just very sensitive to sugar, and so I have to monitor more often. And so I'm essentially the historian in my family, and I gather all the information, and I am very frank and candid in my family. I'm lucky that I have a family that is very candid about the things that run in the family. I understand that that's not always true for every family. But what you can do as the historian is, especially if there are family members that aren't exactly happy to talk about these things, is, you know, you can come back later when there is one-on-one and you can talk and you can share the importance, or maybe you heard something, and you can talk one-on-one and see how that information can help. But I wouldn't push it too much, but I think the first thing to do is to just share how knowing the information could help either yourself or your family members. And so you want to ask if it's okay to ask questions. You want to write down as much information as you can. You want to collect this at least 3 generations. So that's you, parents, children. If you don't have any children, you, parents, grandparents, you, parents. If you don't have any children, cousins, aunts. So at least 3 generations. That can be really helpful in understanding what's going on in the family. One way to get information is, I love Ancestry.com. And so you can actually download death certificates. And so I didn't know that my grandfather had died of a brain hemorrhage. I thought that he had just died, and so in his sleep. And so I didn't know these things. And so it's amazing the type of information that you can actually get from death certificates and from places like Ancestry.com. Type of information to collect: name, the relationship to you, age range, health problems, place of birth, places lived, lifestyle habits, the age at the time of death, cause of death, and whether the death is unknown. And so, for example, I had a friend who was from the Caribbean, and so I think it was a grandparent that died of lung cancer, and one of the things that she was able to find out was that there was a sugarcane plantation, and there was a lot of ash in the air, and so that could have led to the cause of death of lung cancer. There are many, many tools out there. You can Google My Family Health Portrait to get some tools to collect this information. You can also talk to your doctor about recommending a genetic counselor. We actually have several genetic counselors in the state of Virginia, and through Virginia Oncology Associates, this is part of your insurance covers genetic counseling, and the genetic counselor will ask you to collect the information, to collect a family history, and they can make an appointment for the blood test. They can bring the whole family together to explain the importance of genetic testing and genetic counseling. And then when the results come back, they can explain the results and interpret the results to you or to the whole family. They will literally bring in a whole family to talk about the results and how to move forward. And so what I'm encouraging you to do from now and forever, but hopefully the reason Thanksgiving and November is Family Health History Month is because that's when we get together for Thanksgiving and for Christmas. And so when you do get together, try talking about your family health history, write it down, but importantly, share it with other family members, share it with your doctor, share it with your genetic counselor. Share this information with your churches or your community groups. And if you're interested in me coming in and doing an actual workshop on how to collect this information, let me know. And thank you so much. And I'll take any questions.
Thank you so much, Dr. Ricks-Santi, for doing this today. It's really wonderful. I seen a great question from someone, from Dana, asking about if there is, what the state of breast cancer is for African-American women in Virginia. So is there a higher rate of breast cancer in African-American women in the state? And is there a discrepancy in mammograms between different races? And if you know of any initiatives to close that gap. And I had a question similar to it because I know you've discussed in the past presentations I've seen you do about the different health districts and the how they're affected by breast cancer. So if you can give a little bit of information about that.
Yeah, so Virginia is similar to other states in that the incidence of breast cancer is very similar. Can you see me? Can people see me, or should I?
I think you can stop sharing your screen if you'd like to.
That's what I'll do, stop share. I talk with my hands. And so the incidence of breast cancer, so that's the rate at which breast cancer is diagnosed, is still similar across Black and white women, with Hispanics, Asians, Hispanic and Asians having a little lower rate. But one of the things that I've noticed is Native Americans in Virginia have a very high rate of breast cancer, which is different than what I've seen. The mortality rate, so the rate at which Black women die is much higher than compared to white women. So we see that across health districts, and there are some districts that have much higher rates. And so there are districts, I think in Northern Virginia, there are Black women have a higher rate of mortality. In Richmond, I think that Black women have a higher mortality rate. And this is also true for prostate cancer in Black men. So it depends on the type of cancer that you're looking at. You're going to have different rates across different health districts. And so whether or not that is associated with the lack of screening, we really don't know. I think VCU is we're working with VCU actually, they're doing a really good job of trying to tease out what could be the causes of these differences in mortality and in incidence. But those are things that we're actually trying to tease it out. tease out right now as we speak.
Thank you. Another question we had was connected to the transportation resources for patients who are participating in clinical trials. I know that we're going to try to connect some resources, but I didn't know if you knew of any resources to help patients with that complicated aspect.
I do not know, and I can't wait to see our resources, especially with, I know that that's something that you guys are working on, and I know that CACV, the Cancer Action Coalition of Virginia, is working to put together a centralized resources, because there are, we hear about these resources all the time. We have people that reach out to the coalition to tell us about these resources, and we definitely want to do better with centralizing and creating awareness around these resources.
And that's something I forgot to mention at the beginning. Lulu and I serve on the Cancer Action Coalition of Virginia board. I think we started at the same time. So that's how we really got connected. Do we have any other questions? I had a question from Barbara about the slides being available on the website, and just a reminder to folks, yes, we are recording this now. We are planning to make that available on our website. We mentioned before, this is our first one, so we're going to be figuring that technology out, but that is the plan, is to make it available to folks who ended up missing this or want to show it to other people. All right.
I know that I understand that this was all pretty broad, and that's why I'm happy to be the first. So hopefully I could introduce different aspects, and we'll have people come behind me and kind of fill in on some of the other things.
Absolutely. Do we have any other questions from audience members? Or Nikki, is there anything on Facebook that you've seen? All righty, so thank you, Dr. Ricks-Santi. Really appreciate your time today. Thank you to our audience on Zoom and Facebook. As we've mentioned a few times now, this fall VBCF is going to be hosting more virtual education opportunities. So if you're not already signed up for our email list, please do so at vbcf.org/vbcf-connection to stay informed. You can just go to our website and click our publications and sign up that way as well. So thank you all again, and have a wonderful rest of your day.
Yes, thank you for having me. We'll see you soon.
Thank you.
Thank you.